So we are starting now. Dr. Ziyad, you want to say something?
No, I was just greeting Dr. Lushola.
Okay. Yeah, so
I hear you saying.
yeah, Dr. Bishala. Yeah, so this is the beginning of microbiology, and we'll be starting with basic bacteriology. Basic bacteriology. So
Can't see your screen yet.
oh, I'm sorry about that. I've forgotten again. Thank you, Dr. Francois. Okay, what about now?
Yes, and I'm recording.
Yes, I am. All right.
All right.
So basic bacteriology. We start by looking at the bacterial structures. So the bacteria is made up of three essential structures: the cell envelope, the specialized structures, and the appendages. So we are starting with the appendages. The appendages just mean things that are just attached to the major structure of the bacteria. What are these appendages? The flagellum, the pilus, or the fimbriae. So take them one by one. The flagellum is made up of protein, and we this is not the first time we are hearing the word flagellum, flagella. It's just like a whip-like structure that allows the bacteria to move. So this is the picture that shows the flagellum. This is it, this coily structure. So it's made up of protein and it allows for movement. So flagellated organisms or bacteria are motile. The next appendage is the pilus. So let's talk about the pilus. Both the pilus and the fimbriae are made up of glycoprotein. Now, before I move from this flagellum can be present in both gram-negative or gram-positive. Any one of them can have it. So, the pilus and the fimbriae, both of them are made up of glycoprotein. They contain glycoprotein. That is, in addition to the protein, we have some glucan component, some carbohydrate component to them. So what's the function of the pilus? The function of the pilus is quite different from the fimbriae. So the pilus is essential for conjugation. So conjugation is one major event in bacterial genetics that allows for transfer of genetic material. So we are going to shed more light about that as we go down in this discussion. So pilus allows for formation of a bridge between two bacterial cells. So one of them that contains the gene to form the pilus will, manifest that pilus, and that pilus will form a bridge that will connect to the other bacteria, so that the bacteria with the pilus can transfer its own gene to the other bacteria. We call that process conjugation. So pilus is essential for conjugation. Fimbriae, on the other hand, is also made up of glycoprotein, but its own is just to adhere to surfaces, particularly epithelial surfaces. So this fimbriae is essential in as a pathogenic factor for some bacteria because it allows the adherence to epithelial surfaces. For example, the most common example that they usually like to test is E. coli causing UTI. So if E. coli is causing a UTI, it's actually due to the fact that the E. coli can express fimbriae that allows it to attach to the transitional epithelium lining the urinary tract. That is it. That is it. So in that situation, it's not due to the capsule of the E. coli that is doing that, no, but the fimbriae that allows its attachment to the epithelium of the urinary tract. Yeah, so we are done with the appendages. Let's go to the specialized structures. Specialized structures are just cells, are just structures of the bacteria that the bacteria tends to differentiate into in response to an environmental stimulus. Specialized. They do some specific function in response to a situation. Now, the only specialized structure that we are looking at is a spore. So what is spore? Spore is just like a non-dormant form of the bacteria. Whenever the bacteria wants to be, you know, less active, wants to be dormant, you know, they assume that spore form. For example, if it finds itself maybe in an area that is very hot, area that doesn't have water, the conditions are not just favorable for the bacteria. They tend to form spores. So Spores contain keratin-like coats, which are made up of DP-cholic acid. So this DP-cholic acid makes it resistant to heat. HEAT, DP-cholic acid. We should not forget that. DP-cholic acid. Although we are going to hear it more than once, so as we keep hearing it, it's going to stick. DP-cholic acid in spores. Another component of the spore is the peptidoglycan. The last one is DNA. Now, the way you can, marry all these components together is to look at this spore as, okay, we've said it, a dormant form. When the condition become favorable, it's going to move from this spore to the vegetative organism itself. And in that, to form that vegetative organism, you need the nucleus, you need the genetic material. That's why within the spore you have your DNA. And of course, when it's going to the organism itself, it needs to form the cell wall. So that is why peptidoglycan is there. So your spore is made up of peptidoglycan, as well as DNA, and most importantly, DP-cholic acid, so that it can resist, heat, prevent damage from heat. And whenever it wants to go back to the normal form, it will have the peptidoglycan to form the cell wall, and of course, the genetic material is not lost because the spore also contains DNA. So it's being surrounded by the keratin-like coat. Don't forget that. It's typically present in gram-positive organism only. It is only gram-positive organism only that forms spore, because it is only gram-positive organism that forms thick peptidoglycan wall that can help them, resist so many things, osmotic damage, and so many things. Don't forget that gram-positive organisms are the only ones that form the spore. What are these organisms? Think about the B and the C's. The B and the C's. So before I move, I want you guys to prepare your mind that we are going to be learning a lot of codes. So, lot of codes. And I was able to come up with some of those codes for you guys, so that at least we can learn and remember some of those things. But the problem with the code is that you have to learn to remember those codes. That's the issue with the code. If you are trying to recall urease positive organism and you don't remember the code, it's as bad as someone doesn't even know it at all. So, yeah, so spore only for gram positive, because it's only gram positive that contains peptidoglycan cell wall. So we've talked about what the spore does to allow resistance against dehydration, against heat, as well as chemicals. So what are these gram-positive organisms? The B's and the C's. What are the B's? The bacillus. So we talk about the Bacillus cereus, the Bacillus anthrax. So Bacillus anthracis can cause anthrax. Bacillus anthracis, it's a gram-positive rod. Bacillus anthracis, which causes anthrax. Bacillus cereus, which causes rice water. You reheated rice. I said rice water. Reheated rice fever or condition, reheated rice issue, or reheated rice diarrhea. That's your Bacillus cereus. Sereus. Then disease, the Clostridium. So you have your Clostridium, existing in soil as spore. That's once the spore get into a wound, that condition becomes favorable for its growth. Then it changes to the vegetative state. So Clostridium, all of them, Clostridium tetani forming tetanus, Clostridium botulinum forming, causing botulinism, Clostridium perfringens causing gas gangrene, Clostridium difficile causing pseudomembranous colitis. So don't worry about them. You are going to know them as we move. So just know that the Bacillus and the Clostridium are notorious for forming spores. That's the way they can keep themselves alive against non-favorable environmental conditions. So let's look at the third structure, the cell envelope. So cell envelope is more like the covering over the bacteria. It has different layers. The outermost layer of the cell envelope is the capsule. The outermost layer of the cell envelope is a capsule. So this is a capsule here. I hope you guys can see this structure that they are showing here, this zigzag thing. Can you guys see it? Hello? Can we see this? Hello, are you guys with me? Hello? Hello? Okay, okay. Yes,
Can
that's
you re-enter? Can you log in and come? Can you log out and log in?
but I think the network is
Hmm?
good. The network is good so that the stuff will not scatter.
Oh,
I think
okay.
Okay.
But you can hear us now, right?
yes, I can.
All right. All right.
Okay. So the outermost layer is a capsule. Yeah. So you guys should not really bother yourself about the component of all these things, but if you want to learn it, you can learn it. So the capsule contains polysaccharide. That's just it. Forget about the discrete and the loose. Just know that it contains polysaccharide. What is the major function of the capsule? The capsule is just to protect against phagocytosis from the macrophages, particularly due to activation from IgG or complement. So the capsule is to protect against phagocytosis from the um the macrophages. Yeah, protect against phagocytosis from the macrophage. But there's a way you can overcome, you know, that barrier, that capsular barrier. So that's when you now need to employ the activity of the immunoglobulin and the complement. So we are going to talk more about that when we get there. Just know that the only way you can bypass that is by the activity of your immunoglobulins and the complement. But macrophages cannot directly attack a cell that is encapsulated. Once you have something that has a capsule, macrophages cannot go and attack it directly. We will shed more light on how macrophages can attack that kind of cell. So that's that about the capsule. Protect against phagocytosis. Capsule is the most essential virulence factor of an organism. Don't forget that point. Capsule is the most essential. That's the most important. Once you have a capsule, you are very strong. But it's not as if the body cannot still take care of that kind of organism. They can, but just involves some specialized mechanisms. So the next layer is a slime layer.
So the virulence factor, the ability of the organism to cause disease.
Yes, to cause the disease and also protect itself from, damage. Yeah.
So it's
So
both an offensive and a defensive weapon. Okay,
yes,
thanks.
exactly. So slime layer. So slime layer. I think the um I think there's a difference even between pathogenicity and virulence. So I think more of that disease causing stuff property should be pathogenicity, because majority of the virulent factors that we'll be talking about here, Dr. Francois, they are protective. They are protective. So let me show you.
Okay, all right.
They are protective. So don't worry, we'll get there. I will check for the meaning of the
Sure.
virulence so that we can qualify it very well. But majority of
Okay,
the
thank
virulent factors that we are talking about are protective. The capsule, the
you.
M protein, the protein A, as well as IgA proteins. We'll get there. Don't worry. You will see it. You will
All
see
right,
it.
thanks.
All right. So the next one is the slime layer, S layer. So this slime layer also contains polysaccharides. The way I want you guys to picture the slime layer is just like a slimy layer, something that slips, something that's formed like a membrane that swims over something. So that's the slime layer. So that slime layer helps to form what we call biofilms. So biofilms are just like membrane, which involves colony of bacteria attaching through a surface. You guys should not forget that. Slime layer helps to form biofilm. Biofilm means like colony over a surface, attached to a surface, fixed to a surface. Now, what are these organisms that can form the biofilms? There are so many of them. We have about 4 different types. We are going to get there. It's down. We are going to see them. But let me just mention some of them. So, for example, we can form a biofilm on indwelling catheters. That is, if you insert catheter into somebody, if you remove it after like a month, you can see some microorganism coating over it. That's the biofilm. So if you look at it with your microscope, you can see coating of microbes on that catheter. With your microscope, you can see them. So they form biofilm. Example of such organisms include your Staphylococcus epidermidis. They can form biofilms over indwelling catheters. They can form biofilm over prostatic heart valves. So once you hear prostatic heart valve contamination or catheters, you are thinking of staph epidermidis. Don't forget, staph epidermidis. They can also say that, okay, biofilms can be formed over um, uh, a lens. For example, people that use contact lens, you can have biofilms over the eye, particularly the cornea, because of the contamination of that lens that you are putting on your eye. So they can form biofilm over that lens, which can affect the cornea. When there's affectation of the cornea, they can develop keratitis. Someone can form biofilm within the lungs, particularly people that are um, um, on mechanical ventilation for a long time. So mechanical ventilators uh, are I don't want to use the word rich, but they contain some organisms, particularly Pseudomonas. So people that are exposed to mechanical ventilators for a long time, they can develop biofilms within their lung, which contains Pseudomonas organism. So people can also develop um, biofilms within their hair. So, and in the process, they can develop otitis media. The kind of organisms that form the biofilms in that kind of region are the non-typable Haemophilus influenzae. So you guys don't worry, we are going to see all these things and it's going to stay better. Just know that once you hear biofilm, what is coming to your mind is the slime layer. And there are some specific organisms that are very notorious for forming this biofilm. Okay, so let's go to the next one, the outer membrane. So after the capsule, the slime layer, then we look at the outer membrane. Outer membrane contains three layers: the outer leaflet, the middle layer that contains proteins, and the inner leaflet. The most important of all these leaflet that you need to know is the outer leaflet that contains endotoxin. So the capsule and the slime layer can be present in both gram-positive or gram-negative, but the outer membrane is only present in the gram-negative. The outer membrane is only present in the gram-negative. The spore is only present in gram-positive. So what are the components of the outer membrane? We mentioned it: the outer leaflet, the protein layer that is in the middle, and the inner leaflet. So what are the components of the outer leaflet? It contains endotoxin. Now, you guys must remember this. The outer leaflet of the outer membrane contains endotoxin. And what are the forms of this endotoxin? They test this a lot. Endotoxin can be in the form of a lipopolysaccharide or a lipo-oligosaccharide. So lipopolysaccharide or lipo-oligosaccharide. That means you are just saying it contains a lipid component as well as a carbohydrate chain. That's what we are saying. The lipid component of this lipopolysaccharide or lipo-oligosaccharide is called lipid A. Lipid A. Now, if we are looking at a lipopolysaccharide, that means they contain both, the O polysaccharide as well as the lipid A. An organism that contains lipo-oligosaccharide that is endotoxin does not contain O polysaccharide component. They contain the O oligosaccharide, no O polysaccharide. So there are some organisms that contain LPS, there are some organisms that contain LOS. We are going to mention some of them. So for the LPS, we are talking about organisms like your E. coli and majority of the gram-negative, majority of them, E. coli, your Kleb, and other ones. But this LOS
Yes.
is only
Yeah, Dr. Toby, can you hear us?
Yeah, I can hear him, but I can't see his screen. Let me go and come.
Yeah. And Dr. Papa, sorry, before you move far, the LPS and LOS that you're saying, I don't really get, like, you're saying one is for, I didn't really get that part. Sorry. Can
okay.
you take a
Both of them constitute the endotoxin. Now, they contain different components. We've said it: lipopolysaccharide or lipo-oligosaccharide. There's a saccharide component, there's a lipid component. So the lipid component is the lipo component is a lipid. The saccharide component is a carbohydrate. Some of them are polysaccharide, that is, it contains multiple carbohydrate chains. Some of them are oligosaccharide, they are not multiple, they are just like single carbohydrate. That, oligo means few, poly means many. Do you get it? Now, there are some organisms that their endotoxin is made up of lipo-oligosaccharide. That is the endotoxin, lipo-oligosaccharide. There are some that it is lipopolysaccharide. Do you get it, Dr. Zuliat now? Do you get it?
Yes, that's it.
Yeah, so what I just want to mention is that there's a single organism that its own endotoxin is lipo-oligosaccharide, and that is Neisseria. You see it in many questions. They will paint a scenario of someone that has a shock maybe meningococcemia or something, and they will ask you, what is the component of this bacteria that is responsible for this manifestation? You will not pick LPS. You are definitely going to see LPS in the option, but because it is Neisseria, it's going to be lipo-oligosaccharide. It's going to be lipo-oligosaccharide. Now, this endotoxin, what does it do? Endotoxin is native for causing shock. That is it. But it is not the endotoxin entirely that is causing the shock. It is a specific component of the endotoxin, and that is the lipid A component. Because we've said it, lipopoly or lipo-oligosaccharide, they contain carbohydrate and lipid. So it is a lipid part that actually causes that shock, lipid A, because it can induce production of TNF as well as interleukin 1. The interleukin 1 causes the fever component of the shock. Interleukin 1, fever. You guys remember that interleukin 1 is one that responds for fever. Okay. We've not done immunology. Yeah. So interleukin 1 is notorious for fever. Interleukin 1 is also known as osteoclast activating factor. So it can also be responsible for why there is bone resorption, especially in conditions that are shared with elevation of interleukin 1. TNF.
So, Dr. Bradley, I get what you're saying now. Some organisms that are endotoxins, they can have either lipopolysaccharide or lipo-oligosaccharide.
Exactly.
Lipid A is one of, is it the LPS or the LOS, or is just lipid A?
So lipid A is central to both of them. The difference between LPS and LOS is the number of carbohydrate chain that they have. That's why we say.
That affects their, but does it have anything to, like, does it disturb their the virulence, or do you understand my question?
No, it
Or we
doesn't.
just have to learn the ones. Okay, for example, you said Neisseria meningitidis has LOSAB, so it's just to learn the peculiarities with the organisms, right? But lipid A is both LPS and LOS.
Exactly.
Is that what?
And
All right, thank
yes, and it
you.
is that lipid A that is inducing that shock. That is the major component of lipid A.
Okay, immunologic. Okay, TNF. All right,
It
thanks.
induces TNF and
Interleukin.
interleukin 1.
Okay.
Exactly. So the polysaccharide component is there too, but it can also be antigenic. So you can have production of antibodies against it. But the major one that drives that inflammatory process is the lipid A, which is present in both LPS and LOS. So now, of course, in the middle layer of the outer membrane, we have the embedded protein. So those proteins contain sporine, and they also contain other outer membrane protein. So those outer membrane protein are just antigenic, just contain the antigenic stimulus for that microbe. That, okay, those are the components that the white blood cells will see and they will react against. That's what it means. It's not as if they induce any inflammatory stuff. That's it. So they are just like the signals, or like the flag that the microbes carry, that, okay, that the white blood cells can see and recognize them. That's the antigenic component. Your outer membrane protein within the um middle layer of the outer membrane. So in the inner leaflet, we have phospholipids. They are just there. Phospholipids are just there. They don't do any particular function. Now, within that middle layer, we said the middle layer contains the outer membrane protein. It also contains porine. So that porine just allows for movement of, cell of structures, or, you know, um, anything. Just allow movement of materials between, the cell and the environment. That's just what the porine does. Porine just allow for movement of materials within that outer membrane. So the outer leaflet contains the endotoxin. What are the components of the endotoxin? LPS or LOS, depending on the organism we are looking at. Now, what particular part of the LPL or the LOS drives the inflammatory response causing shock? It is the lipid A. So the lipid A triggers release of interleukin 1 and TNF alpha. Interleukin 1 causes fever, TNF alpha causes fever as well as hypotension. So it mediates both fever and hypotension, while interleukin 1 is only fever. Both of them are part of the component of shock. You guys remember. Then, embedded between the outer and the inner leaflet, you have the middle layer that contains embedded protein. What are these proteins? Porins. Porins allows for movement in between. Then we also have the other outer membrane protein that are antigenic. So that's all about the outer membrane. Then once we move beyond the outer membrane, and you remember, outer membrane is only found in gram-negative. So that is why it is only gram-negative that contains endotoxin. Don't forget that. Gram-negative. So next to the outer membrane is the periplasmy. The periplasmy or the periplasm. So the periplasm um, is a space between the outer membrane and the cytoplasmic membrane, not cell wall. The periplasmy is the space between the outer membrane and the cytoplasmic membrane, not the cell wall. So what does it contain? It just contains hydrolytic enzyme. So enzyme that can help to digest materials within the cell. What are these hydrolytic enzymes, particularly the beta-lactamase, the beta-lactamase. That's it. We'll talk about this beta-lactamase now as we are progressing. So the next, after the periplasmy or the periplasm, why am I finding it very difficult to pronounce this thing? Periplasm, the periplasm. So after the periplasm, we have the cell wall. So this cell wall contains peptidoglycan. Peptidoglycan is present in both gram-positive and gram-negative. Cell wall is present in both gram-positive and gram-negative. But the difference is that the cell wall of the gram-positive is thicker. It has a thicker peptidoglycan cell wall than the gram-negative organism. What is present within that peptidoglycan? From the name, glycan peptide. So they have carbohydrate backbone that contains side chain, that are protein, that contains protein elements. And we have, cross-linking of those proteins by peptide bonds. So when we say cell wall is made of peptidoglycan. The cell wall of a gram-positive is more developed, is thicker. It has a thicker peptidoglycan compared with the cell wall of
Dr. Babawale.
yes, Dr. Rishala.
So a quick question. So does that mean the cell wall of gram-negative is also inside the periplasm?
No. I said periplasm is a space between the outer and the cytoplasmic membrane. So that means that the periplasm is just above the cell wall. Do you get it?
Because cell wall is in between now.
Yes.
It looks in between the peptidoglycan layer is in between the two of those places you talked about.
Okay. So what is now your question now?
I'm asking if cell wall is in between them.
Yes. I said periplasmy or this outer membrane is present in gram-negative only. Do you get it? You don't have this periplasmic space in the gram-positive because they don't have outer membrane. Do you get it?
Okay.
They don't have outer membrane. So let's look at the image. So, this is the periplasmic space. Although we can have something that looks like it here, but the best qualification for that space is actually the gap between the outer membrane and the cell membrane or the cytoplasmic membrane. But you know that the gram-positive, they don't have outer membrane, so it's not you can't unless you are saying that okay, maybe it's between this structure here, which is your cell wall, but it's not really something that we should worry ourselves about. So yeah, so that's why you see it here that they said it specifically that it's a space between the cytoplasmic membrane and the outer membrane in gram-negative, so with the peptidoglycan in the middle. So it's more for gram-negative organisms. Yeah. So for gram-positive organism, the cell wall is thicker. It contains a thick portion of the peptidoglycan, and that peptidoglycan cell wall is just to give it support, protect it against osmotic damage. That's what it does. So the take home here is that peptidoglycan cell wall is made up of both carbohydrate and protein components. So you have cross-linking of the protein part by transpeptidase. So transpeptidase is an enzyme that allows cross-linking of the peptides of your peptidoglycan. So you can have drugs that can act against this transpeptidase. So that's the mechanism of giving penicillin. So if you give somebody penicillin, you are inhibiting formation of this peptidoglycan cell wall. And you know that peptidoglycan cell wall is more predominant in gram-positive organisms. That is why our penicillin can work more for gram-positive organisms. We still have little peptidoglycan in the gram-negative. So, and of course, vancomycin also works, through this mechanism because vancomycin can inhibit dealanine. So dealanine is like an amino acid, that is cross-linked. So it inhibits formation of that dealanine, so that you cannot eventually have any cross-linking by your transpeptidase. So we'll talk about it when we get to the drug text section. But I just want you to always remember that penicillin inhibits transpeptidase activity that allows for formation of peptide bond between the protein, structures of our peptidoglycan. And the peptidoglycan cell wall is to provide rigid support, protecting against osmotic damage. So if you place it in an hypertonic or hypotonic cell, it doesn't get burst because of the peptidoglycan. So the last layer is the cytoplasmic membrane. So that is the one that is, leading to the interior of the cell directly. So this layer here we are seeing here is a cytoplasmic membrane. This area here. So the cytoplasmic membrane is actually the cytoplasmic membrane contains phospholipid bilayer. So the way the phospholipid bilayer are arranged is that the heads, which are the polar region of the phospholipid, are facing outwards. Some are facing outwards, the other ones are facing inwards. So that the hydrophobic tails are the ones in the middle. So you can easily have penetration of, you know, lipid soluble substance through this phospholipid. They can enter because the area that is inside and the area that is outside are both containing the phobic, so that, sorry, I said I am lipophilic, hydrophilic substance. So hydrophilic substance can easily enter from inside or from outside because the heads of the phospholipid, molecule are facing outside and they are facing inside, while the hydrophilic tail are the one in the middle. So that's where the cytoplasmic membrane is oriented. It contains phospholipids. That's essentially what it is. But within these phospholipids, you can have proteins being embedded within them. So part of this protein includes what we call the penicillin binding protein. The penicillin binding protein. So this penicillin binding protein have some enzymatic activity. Part of the enzymatic activity that they exert is transpeptidase activity. So within this phospholipid, you have proteins that we call penicillin binding protein, which have some enzymatic activity, which includes transpeptidase activity that forms the cell wall that is just next to it. So they help in forming the cell wall. Then within the cytoplasmic membrane, we also have some projections that we call the lipoteichoic acid. So lipoteichoic acid is only native to gram-positive organisms. You only find it in gram-positive organisms, and it is there to induce, shock-like presentation. So the TNF expression as well as interleukin 1 from infection from gram-positive organism is actually due to response to the lipoteichoic acid that we can see here. Yeah. So penicillin binding proteins are, the proteins that our penicillin binds to. So when you give somebody penicillin, it's going to bind to that penicillin binding protein and it will inhibit them from functioning. And we remember that part of the function of the penicillin binding protein is what? Transpeptidase. And transpeptidase allows for formation of a peptide bond between the amino acid, forming the peptide component of our peptidoglycan. So also within that cytoplasmic membrane, we also have some enzymes that allows for transportation as well as oxidation of molecules within the cell. So what I just want you guys to remember within the cytoplasmic membrane is, okay, it has phospholipid bilayer. The polar head are facing outward, the other one is facing inward. Submerged within them is our penicillin binding protein that has enzymatic activity, and also some enzymes are also there. Apart from synthetic activity of the cell wall, some enzymes are also there that allows for oxidation as well as transportation. So then lipoteichoic acid induced interleukin 1 and TNF alpha. So that's that about um the the structure. So you can see this peptidoglycan cell wall, this blue component, you can see that it's thicker than this guy that only contains one. And you can see the structure that makes up the outer membrane. You don't have that kind of structure here. You don't have that kind of structure here because it is only present in gram negative. It is not present in gram positive. So after the capsule, you have the slime layer. After the slime layer, you don't have the outer membrane for the gram positive. The next is the cell wall. Then after the cell wall, you have the cytoplasmic membrane. But the gram negative, you have the appendages that are there. Then the the capsule. Then after the capsule, you go to the slime layer. After the slime layer, we have the outer membrane that's made up of, endotoxin, as well as the proteins in between and the inner phospholipid. Then, after the outer membrane, the next thing is the cell wall. Although between them, you have the periplasm that contains, some enzymes like the beta lactamase. Yeah. So after the periplasm, you have the cell wall. Then after the cell wall, you have the cytoplasmic membrane. So that's the way. You guys might not remember the attachment. Don't worry yourself about how they follow. It's not really important. They don't test that. What is important is what they do. You guys will be able to remember what they do. So what is the component of the cell wall? What does it do? What's the component of the cytoplasmic membrane? What does it do? What's the component of the outer membrane? What does it do? What's the function of the slime layer? What does it do? Capsule, what does it do? Don't worry yourself about the arrangement. If you don't remember arrangement, nobody will ask you which one follows. The most important thing is you remembering, you know, the component of each of them, particularly the cell wall and the outer membrane, and what they do. Nobody will ask you capsule, what does it contain, like polysaccharide. That's useless. That's not information. But cytoplasmic membrane contains penicillin binding protein, which can be inhibited by some drugs, and that penicillin binding protein exerts enzymatic activity like transpeptidase. Our outer membrane contains endotoxin, which can be LPS, LOS. That's information, very important because it's pathogenic. They can induce production of, TNF and interleukin-1 from their lipid A component. That's question. Lipotechoic acid is only native to gram-positive and it release interleukin-1 and TNF alpha. Very important. Very, very important. Okay. So let's go to the stain. So we have so many stains. The one that we are most familiar with is the gram stain. So gram stain, what we just do in gram stain, we just get the bacterial cell, we make the smell on our slide, we had crystal violet, which is a premise stain. So after you add crystal violet, you add Lugos iodine. So Lugos iodine is there to adhere the crystal violet to the cell wall. Now, once you add the crystal violet, if the bacteria contains a very thick cell wall that contains abundant peptidoglycan, they are going to take that crystal violet. They are going to take it up. So, for example, a gram-positive definitely will take up the crystal violet. So, if you add the Lugos iodine, it's going to add that crystal violet stain to the cell wall. It will make it adhere very well. So, look at your iodine as a fixating agent. Nobody will ask you this. I'm just trying to explain so that we can understand the principle. So your Lugos iodine is there to make it adhere to the cell wall, the crystal violet. Then subsequently you add a decolorizer. The reason why you're adding a decolorizer is that, okay, let's say that this bacteria is not a gram positive, is a gram negative, and it contains little um little peptidoglycan. So once I had, my crystal violet is going to take little crystal violet, and if you add iodine, there's little cell wall for the crystal violet to adhere to. So that means that once I had the decolorizer, it will wash out both my crystal violet and my iodine. Everything will go out. But if the bacteria is a gram-positive bacteria, once you're adding the crystal violet and the iodine, everything will merge with the peptidoglycan. If you're adding a decolorizer like alcohol or maybe an acid, it will not be able to wash it out because it's already fixed to the peptidoglycan. So, subsequently, after adding your decolorizer, if you now had a counter stain like safranin, because it has taken up the crystal violet, is not washed up by the decolorizer, it will not take up your safranin. So it will still remain the color of the crystal violet for gram positive, and that is a purple color. Very easy. So if it's a case of a gram negative, if you add your crystal violet, little is obtained by the cell wall. If you add iodine, there's little cell wall to attach the crystal violet to. So it will not really attach. Once you add your decolorizer, it will wash out everything, which is alcohol or acid. It will wash it out. So by the time you're adding your counter stain, which is safranin, that is red, it will only take, the color of the counter stain. That is why a gram negative organism will appear red, while a gram positive organism will appear purple. So what are the problems of gram staining? What are the organisms that don't follow this gram stain principle? An organism that is too thin to visualize. You cannot see them with your life because what you do with your staining is to look at them in your light microscopy. So you can even barely see them because they are too tiny, particularly your treponema and your leptospira. So you are talking about the spirochetes. So spirochetes generally are too little. They are even they are too small to be seen. So that's why you don't use gram stain to look at them. You use something more sophisticated, like maybe you use your fluorescent antibody test, or you do a dark field microscopy. That one is more powerful than gram stain. So we should know the reason why you cannot appreciate these guys. Your leptospira and your treponema, the spirochete generally, they are too thin to be visualized. That's why we don't appreciate them using gram staining. Mycobacteria is not gram stain because of its high lipid content. So that high lipid content doesn't allow you to take up, the stains very well. So you have to use something else. The high lipid content of microbacteria, it contains very high amount of mycolic acid, makes them difficult to pick up the stain. Mycoplasma, ureaplasma, these guys are like atypical organisms. We are familiar with them. They contain no cell wall. So because they have no cell wall, there's nothing to pick up the stain. They are not visualized by, gram staining. Mycoplasma, ureaplasma. Then the last one are the intracellular organisms. So I think the code for the intracellular organism, what's even the code now? Can I remember? I have to write all these things down. What did I even say? Oh my god. Okay, I think I used the word crypt. I think I used crypt. Okay, that code might not work for you guys, but that's what I could come up with. Crypt. So CRPT. So you can put the Y there, but just know that the Y doesn't stand for anything. But my problem with using the Y is that you might think Y is Yersinia. So it's either you just spell the crypt as CRPT, crypt HB. So I was able to include the HB, thinking about HB visa
Which code is that?
for the gram
As in,
stain.
I'm trying to from the what?
Is
For the
it
what?
Oh, sorry.
actually that's not James Sir, that's James
I'm,
Sabi.
I'm already talking about
Ah,
James. I'm sorry.
Okay.
because the one that we are seeing is
I'm sorry.
Legionella, Rickettsia, and the likes.
Yeah.
Those
So
are the intracellular organisms.
yes. So let me see what I use for those ones. Uh, okay. Yes, I remember. So what I use for those ones is BCR ABL. Do you guys remember what BCR ABL means?
Yeah, Philadelphia chromosome.
Exactly. So what you just do. So it's difficult for me because I could not add note to this thing. So I will appreciate if you guys can just write it somewhere so that you guys can remember and maybe also help me remember it. So BCR ABL. Now that ABL make it Abel of the Bible. So the B
Okay,
is
that
standing
makes
for
sense. So
the
it's
B exactly. So the B is
and
standing for the
Legionella.
yes,
Okay,
the B.
okay.
But anytime you see chlamydia, most of the time the catcher will follow it. They go together. Those guys like themselves. They are like very similar. Sorry guys, 1 minute. Okay guys, yeah, so BCR ABL ABEL. So the B is standing for the Bartonella, the C is standing for chlamydia, and the next thing is the R, which is Rickettsia, then the A anaplasma, the E is Ehrlichia, then AB. We've talked about the B, right? So Bartonella, then the E Ehrlichia, then the L Legionella. So these guys are not only intracellular, they're also very small. And one of them, that is chlamydia, contains little muramic acid. So muramic acid is also one of the components of peptidoglycan. So it's part of the carbohydrate component, muramic acid, N-acetyl muramic acid. It's part of the carbohydrate component that the proteins join with to form the peptidoglycan. So because chlamydia has little muramic acid in them, so that the cell wall of chlamydia is not well developed. It's not that it contains no cell wall, it just contains a poorly developed cell wall. While the other ones are strictly intracellular, so it's very difficult for the stain to get into them, and they are very small. So that is the code. I hope you guys will be able to remember the code. BCR, BC, BCR, ABEL, right? Dr. Luatobi, are you still there?
Yeah. BCR Abel.
Yeah. So the most important thing is you being able to realize the reason why they call it.
BCRAB, like BLABL. So it makes sense. Thanks.
Yeah. So you guys must be able to remember why you don't appreciate them on gram state, not you being able to remember them only. Why are they not? So your spirochete, they are too small. Mycoplasma has mycobacteria has too much lipid. Mycoplasma and ureaplasma has no cell wall. You have seen the application of this mycoplasma and ureaplasma having no cell wall. We solved the question. You guys remember that the answer was
I was
mycoplasma.
saying my
Yeah.
sister.
Exactly.
Yes.
You can
Yeah.
see
That
now
she was
that
confusing mycoplasma for mycobacterium.
exactly. So you can see it now that that's the reason. No cell wall. Then
So
the BCR ABL. Okay. So the next one is the GIMSA. You see, one thing about this GIMSA, the way I master this GIMSA is that I picture, you know, whenever I hear GIMSA, I think about the protozoa. Think about the protozoa. You will never be wrong. You know, it's good to know all these things. I cannot lie to you guys. Before I went for my exam, I'm not sure I could remember all of them. I knew them, but it's not as if I can record them spinal level. Like I didn't code anything. So I just learned the one I could learn and then just went for it. So, but if you guys want to learn everything, it's fine. Just want to, dot all your I's, cross all your T's so that you don't want them to get you from any angle, which is very good. So, James Stain, think about the protozoa. What are the protozoa that we are talking about? Trypanosomes, Plasmodium. And we've already said it that whenever you see chlamydia, rickettsia follows. Now let's now use that code. So the code is what? Crypt HB. Now, Crypt HB. HB is one of the visas that every international medical graduate is hoping to get in the US. HB is a work visa. So once you have a HB visa, you can get your green card within 5 years. Everybody likes the HB visa. So the HB of the HB visa is standing for Helicobacter pylori, and the B is standing for Borrelia. So Borrelia is a form of spirochete. We should not forget that. Borrelia is not, this B in this GIMSA is not Bartonella. It is not Burgidophilia. It is Borrelia. So Borrelia and Helicobacter, that's the HB. So what's the crypt? Chlamydia. Once you remember C, we remember the R immediately. Chlamydia, rickettsia. Then we ignore the Y. Then PT, Plasmodium, Trypanosome. Plasmodium, Trypanosome. Those are the protozoa. Plasmodium, Trypanosome. So these are the way.
Dr. Baba, how did you spell the script? How did you spell the script?
C R.
CR.
You know,
Crypt of Libacum.
exactly. You know that exactly. So you know that crypt is normally CRY. So it's now CRPT.
Mm-hmm.
If you want to put the Y, you can put it there. Just know that the Y doesn't code for anything. Don't go and call the Y a Yersinia. So that's my fear with
Okay.
the
So is it crypt HB or what?
crypt HB visa. Crypt HB. That's it. Crypt HB.
Okay.
Crypt HB. Yeah. So most of the time they will just test for trypanosome and plasmodium. That's the major thing for this GIMSA. Trypanosome and the plasmodium. They commonly use it to stain them. They are the protozoa that are there. Now, this is where it appears. So once you see a flagellated stuff like this with red blood cells there, you know that that's likely to be trypanosome. Trypanosome. This is GIMSA stain for trypanosome. Okay. Um, Periodic active SNP. So PAS is majorly used for glycogen. It stains glycogen. So it also stains, mucopolysaccharides. So glycogen storage disease, people with mucopolysaccharidosis, they can have stained by PS. So glycogen storage disease can be identified with PS. Mucopolysaccharidosis can also be seen with our PS. If you want to stain macrophages infected with Trephorema wipae, you're also going to see with the PS. There are so many organisms. See, we can't begin to be listing them. It's a lot. Until you have a one antitrypsin, so many diseases can be seen with PS. But let's just stick with these guys: glycogen, mucopolysaccharides, as well as Whipple's disease. So Whipple's disease, because this Treporema wipae can get engulfed within macrophages. So these macrophages takes up these PS, so you can see them. If I ask us how Whipple's disease present, I'm not sure can we remember Whipple's disease? How did they present Whipple's disease?
They have diarrhea, they have
Okay.
steatorrhea,
Abdominal pain,
they have abdominal
malabsorption syndrome,
cramps, yeah, malabsorption. Then it can go to, I think it can affect their heart
then the
too. I'm
joints,
not sure.
Fantastic.
then the
That's it.
joints.
That's it.
I'm not
The
sure.
joint and the heart. Yeah. Can affect the heart, can affect the joint. That's it. Then diarrhea. Treponema with pain. So that's the way it looks like. This is it. This is PS. This is the way it looks. You see all these, blue, violet looking organisms. That's the way it stains. That's the PS, periodic acid SNP. Okay. Then the next one is the senescent stain. So senescent stain is for mycoplasma, for acid fat organism, not only mycobacteria. Senescent is for acid fat organisms. That is organisms that you cannot counterstain with acid. They are resistant to counterstaining with acid. What are these organisms? The mycoplasma, the mycobacterial tuberculosis, mycobacterial avium, mycobacterium leprae, any of the mycobacterium. Then you also need to know the other ones. Nocardia is also partially acid fast. Nocardia. Some protozoa like cryptosporidium. So you guys must learn today that it's not only mycobacteria that is acid fast and that we can stay with the senescent stain. It is not only mycobacteria. So the component of the senescent stain is the carbonfourteen. It is not only mycobacteria. Nocardia is also acid fast. Some protozoa like cryptosporidium that causes diarrhea, particularly in people with which condition? Anybody? I'm just throwing it out. Anybody will know?
HIV,
HIV.
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 AIDS, yeah.
Fantastic. People with AIDS can have cryptosporidiums. Then isospora, isospora belly, cyclospora, all of them, they are acid fast organisms. So if you don't have access to this Zenesis stain, which is actually very good, you can use oramine rhodamine stain. So it's very sensitive and it is cheap, compared to Zenestin stain. And it's very sensitive too. Yeah. The next one is Indian ink. You see this picture a lot. So this is how our acid fast stain, this Zenestin stain looks like. You see purple looking organism in a blue background like this. Once you see this, you know that this is likely to be Zenestin stain they are talking about, acid fast organism. This is the way it looks like. You have to be familiar with these pictures because you are going to see something very similar in questions. So the next one is the Indian ink stain. Indian ink stain is very conspicuous. This is what you see. Just see white with chocolate background. And the stuff will be looking circular. These are the capsules. It's highlighting the capsule of the organism. Indian ink stain, it's only used to stain Cryptococcus neoformans. That is the only thing. If you don't remember anything about Cryptococcus, remember Indian ink stain. And this is what it looks like. Indian ink stain, Cryptococcus neoformans. And it is because Cryptococcus neoformans is highly capsularized. It contains a very thick capsule. That is why it can cause serious meningitis. The white blood cells find it very difficult to destroy it because of that thick polysaccharide capsule. So this is the way it appears, white with like a chocolate background. So if they don't use Indian ink, sometimes they can say they used musicamine. So they can say they use musicamine stain, which makes it appear red. So with musicamine it will appear red, but with Indian ink it will appear like this. So don't forget, Indian ink, musicamine are both for Cryptococcus neoformans. Don't forget. And Cryptococcus neoformans, its virulence factor is a very thick polysaccharide capsule. Guys, you can see that I'm emphasizing this thing. Please, I want us to pay attention to it so that I can be happy when we are solving questions and we can remember all these things. I'm not just saying it because I just want to say it. Those are the important ones that we need to know. Cryptococcus neoformans, thick capsule, and the stain, musicamine, and Indian ink. Cryptococcus neoformans. Remember meningitis. Okay. Silver stain. So once you guys see silver stain, fungi is enough. If you want to learn the other one, it's fine. But majority of the time, they test it with the fungi. What are the fungi particularly that they like? Pneumocystic, Aspergillus, coccidioidosis organisms. So coccidioidosis, Pneumocystic, Cerevisiae, and Aspergillus. They can even use to stain Candida, can stain any of the fungi, silver stain. and it appears green. So once you see something that appears green like this, that's silver stain. Silver stain appears green. You can also use it for the other ones, like the Helicobacter, Legionella, and Bartonella. So lab, I think I use lab to code that. So once you see silver stain, there's no code for the fungi. Just remember fungi. Silver is fungi. That is it. It's just like Saboroid agar. Saboroid agar is to culture fungi. That's the agar to culture fungi. The stain to visualize fungi is silver stain. But the other thing that we can use it for includes the lab. What is the L? Legionella. What's the B? Bartonella. The A is looking like H. Just change it to Helicobacter pylori, lab, and fungi, silver stain. Okay. Then the last one is a fluorescent antibody stain. I've said it that there are some organisms that you cannot see on the light microscopy with gram staining. So some of them include our spirochetes. If you want to look at your spirochete, you want to use fluorescent antibody stain. So antibodies against those organisms are seen, and you stain them. It's a molecular technique. So fluorescent antibody stain for syphilis, you can use it for pneumocystic, you can use it for Giardia. It's not very high yield. You guys, it's very common for syphilis. Don't bother yourself about this Giardia. Nobody will ask you. But syphilis, you must remember, fluorescent antibody stain to check for syphilis. Very, very important. We are going to shed more light on that when we get, to syphilis. Yeah. So, what are these culture medium that we can use to grow our organisms? Yeah. So, we have to know all these culture medium to very well. They are high yield. Haemophilus influenzae is cultured in chocolate agar. So, chocolate agar is a form of enriched agar. So, enriched agar just means that it contains nutrients that can facilitate the growth of that organism. So, if you have 20 organisms that depend on factor 5 and factor X to grow, factor 5 codes for NAD+, factor X is hematine. So, if you have 100 organisms that can grow in a media that contains these two organisms, that is and you put them in the media and they grow, that is an enriched agar. It doesn't discriminate between one. It doesn't kill off one for another. It will allow all of them to grow. And what is present within that media is actually chocolate. So you put blood. It's more of blood. So blood that looks like chocolate. So it's more of blood. It's blood that is there. It looks chocolate colored. Yeah. So chocolate agar contains blood majorly. All these factors that you are seeing here are present in the blood. Factor 5 and factor X. So factor X known as hematine, factor 5, releasing NAD+. Yeah. So you guys must master these two guys as component of chocolate agar, which allows the growth of Haemophilus influenzae. Another agar is what we call the Thiamartin agar. So Thiamartin agar is a form of modified chocolate agar. That is, we are converting this chocolate agar from an enriched agar to a selective agar. That is, even though we know that 100 organisms can go in this chocolate agar, we want to make this chocolate agar grow only one organism. And the only one organism that we want this agar to grow is Neisseria. How can we do that? Put in some antibiotics or antifungal agent that can kill off other organisms that can grow in the chocolate agar. Once you add antibiotics and other, drugs to chocolate agar, you convert it to the Thiamartin agar. It also contains chocolate with the I said chocolate blood within it. So it's more like a chocolate agar with antibiotics embedded in it. So it selectively favors the growth of Neisseria, making it a selective agar. So you're not going to pick enrichment agar in that kind of situation. It is a selective agar. What are the things that destroys the other organism? It contains vancomycin. So what organism do you think vancomycin will be working more against based on what we have talked so far, what we have said so far?
That would be gram positive.
That will be gram positive. Why?
Yeah. They have the cell wall.
Exactly.
So they have the cell wall.
And
so it will bind to the DLR part of their peptidoglycan layer.
Exactly. Thank you, Dr. Oluwatobi. So it affects formation of growth of gram positive organism, vancomycin, because it inhibits the formation of the cell wall. Yeah. Gram negative organism, you can take care of that by using trimethoprim and cholecystine. Who remembers the mechanism of action of trimethoprim? Anybody?
Hydrofolate reductase
You are moving stuff.
inhibitor.
You are moving stuff, Dr. Latobi. Hydrofolate reductase in bacteria. The hydrofolate reductase in protozoa. What is that?
Pyrimethamine.
Perimetamine. Perimetamine. Thank you, Dr. Latobi. Then the last one is cholecystine. Cholecystine or polymyxin. Any one of it. It can be used interchangeably. Cholecystine or polymyxin. Alongside with the trimethoprim, they inhibit the gram-negative organisms. Then the last one is the fungi. So how do you want to take out the fungi? Use nystatin. So nystatin will inhibit the growth of the fungal agent. Yeah. So that's that about the Martin agar. The next one is Bordetella pertussis. So how do you want to grow Bordetella pertussis? From the name, you can get it. BP. So B, Bordet gengu. Bordet gengu. You guys have, this is the first time you are seeing it, so you have to be able to familiarize yourself with it. I don't expect you to remember it today, but with time you get used to it. Bordetella pertussis, Bordet gengu. So because of the P, the B is already standing for the Bordet gengu. So the P part contains what it talks about, what it contains, potato. Nobody will actually ask you what it contains like that because it's not really very high yield. But if you want to know it, it's fine. That's the code. Pertussis, the P, potato extract, potato. Then the second one for the Bordetella pertussis is the Regaloa medium. So Regaloa medium contains ABC. A, antibiotic, B, blood, C, charcoal. So Bordetella pertussis, Bordet gengu. The P, pertussis, potato extract. The second one is the lower regal. Lower regal contains ABC, antibiotics, blood, and charcoal. This one is very high yield. Corynebacterium diphtheriae, the medium, we must know it. Two major mediums, the Loeffler medium and Telluride agar. They had this one, the standard stuff, you must know it. In the Telluride agar, you are going to see black colonies of Corynebacterium diphtheriae. Telluride agar, black colonies. The Loeffler medium, you are going to see metachromatic granules or granules that looks like club. You guys remember Orogun? You guys know how Orogun looks like? Hello, guys. So, hello? Do you guys
Yeah.
remember I
Yes.
mentioned Cryptos when we were talking about them?
Mm-hmm. Yes.
Oh my God, I forgot to resume recording. Ah, yeah, so that Cryptos is due to the fact that they produce gas, that hair that settles within the subcutaneous tissue. Yeah, so you guys should not forget that. It's difficult to culture them because you need an anaerobic environment. So you need sophisticated equipment to be able to create that kind of environment. So that's that about them. And anaerobes typically are normal flora of the GI tract. So it's not when you are now having anaerobes in other part of the body, like maybe the skin, maybe the mouth, or other region that you now start having problems. So another thing about anaerobes is that antibiotics like aminoglycoside cannot work against them because for a bacterial cell to take in aminoglycoside, they need oxygen. So they need oxygen to take in aminoglycoside. So a bacterial cell that does not utilize oxygen cannot be affected by aminoglycoside. So aminoglycoside doesn't work against anaerobes. We should remember that. Yeah. So what are the facultative anaerobes? We are talking about organisms that
You
can.
said antibiotics that doesn't need oxygen.
No.
Or how did you put it?
Bacterial cells that do not use oxygen cannot be affected by aminoglycoside because you need to for the aminoglycoside to enter that bacterial cell, there's a need for oxygen to be present within that cell. Do you get it?
Not
Hello?
yet.
Yeah. So if there's no oxygen, there's no way the aminoglycoside can be taken into the cell. So there's no way it can work.
Okay.
So it doesn't work against anaerobes. Yeah. So facultative anaerobes are organisms that can, behave like normal aerobic organisms and they can behave as anaerobic organisms depending on the situation where they find themselves. And you guys remember when we were talking about the electron transport chain, that's a condition that can happen in the presence of oxygen because the electron transport chain is also known as oxidative phosphorylation. So oxidative, you need oxygen. And what they do is that they accept the electron. The electron has been transmitted in the electron transport chain. Some of them get transmitted to oxygen. It will convert the oxygen to water. So you transfer electron to oxygen, you get water. You get water. So, and they use that oxygen accepting electron to generate energy in form of ATP. So, they can do that when there's oxygen, and if there's no oxygen, they can go through the anaerobic pathway and they'll be fermenting their polysaccharides and their carbohydrates through oxygen independent pathway. So what are the organisms that can behave this way? Strep, staph, and our enteric gram-negative bacteria like E. coli, like Kleb, and the other ones. Yeah. So don't forget the word facultative. It means I can switch aerobic and aerobic. So what are the cells that are strictly intracellular? There are ones that are obligate intracellular, that is, they cannot exist outside the cell. Once you move them out of the cell, they die. So rickettsial and chlamydia, Coxiella, yeah, but predominantly rickettsial chlamydia. Once you remember this, very easy. You hardly test that for Coxiella, but rickettsial chlamydia, they follow each other. Obligate intracellular. They don't have any choice. They must always be intracellular. So the facultative intracellular, they can work both outside and inside. So they can stay inside and outside. So that's these guys here. And I think we have a code for them. MBME slide. So that's the code I came up with. Although me, I know that all these things nobody will ask. I'm just giving you the code so that you guys will not say that we are just skipping over them. I'm sure nobody will ask you facultative intracellular. Nobody. So, but if you want to know it, the code is SLY MBME. So MBME sliders, MBME slide. So just replace the E of MBME with F. That is all. That is it. So MBME, the M is the Neisseria, the B, MBME brucella, the M Microbacterium, the E is the Francisella. You replace it with F, Francisella, then SLY, Salmonella, Legionella, Yersinia. So do we get the code?
Yes.
Yeah.
Dr. Babawale, so
Yes,
how come because I'm trying to correlate Coxiella and the gram stain. So Coxiella will not stain gram, abi?
yeah, it doesn't stain gram. It will not. It will not.
Because I'm trying to bring all of them together. The obligate intracellular and the intracellular from gram stain, the BCR one. You
Yeah,
get
particularly these obligate ones, they will not stain. The facultative can still stain. They can both be inside, they can be outside.
because
You get?
Neisseria is gram negative.
Yeah. Those ones are facultative. Do you get? They don't
Okay.
stay strictly inside. Sure you get?
It depends, abi.
Yeah, so they can be outside, they can be inside. So, once they're
Okay.
outside, you can if you culture like, if you culture like let's say like an infected stuff, and you are trying to stain for Neisseria, it's not everything that will be inside. Some will be outside, so you can easily, culture them. So, and when we say they are inside, that means they're like inside like macrophages or something. But if you have extracted them out of, how do I even explain what I'm saying? Like maybe you have an infection now, so you now put it in the growth media. You know that you are going to grow that organism, like say within that growth media, and it will not be growing inside
Mm-hmm.
macrophages because it is not inside the body again. You have brought it outside. Do you understand? So do you get what I'm saying now?
Hmm.
So
I'm trying to get it. I'm trying to get the facultative is like when they are within the cell, you would be able to stain them. But when they're outside the cell, you might not be able to. Like, it is when they want to infect the cell, they have to go inside the cell when they want to infect the cell. Is that what you're saying?
yeah. So this is it. When you use the word facultative, that means that we are trying to say in between. It is neither there or here. That means they can do both. It's just like facultative anaerobes. They can both use oxygen and they can survive in the absence of oxygen. It will not exclude.
Exactly.
Now,
Yeah.
when we say facultative intracellular, that means you are saying that it can survive inside the cell or outside the cell.
When they are facultative.
Yes,
Okay.
facultative.
Obligate, they have to be inside the cell, Nisha.
Yes, they have to be
Okay.
inside the cell. That is it. So it makes sense for us to be able to gram stain an organism that can both survive inside the cell and outside the cell. You see?
Yeah.
So
Yeah.
that is it.
I think it makes sense. Thanks.
Yeah. So encapsulated organism bacteria, this one too is very high yield. We have to know them. But the common one that they commonly repeat is the shin. SHIN. So the shin, the S there is a streptococcus, the H is Haemophilus, HI, Haemophilus, then the N, Neisseria, shin. By the time we go find this microbiology, you will not even need to be using code again because you already know these guys, the encapsulated one. Once you get encapsulated, you are thinking of strep, Haemophilus, Neisseria. Those three guys, they move together. They cause a lot of things together. They have so many
Okay,
common.
Dr. Brawley, sorry, please hold that thought. Just before you leave. So encapsulated from the bacterial structure means that
Mhm.
capsule within their cellular wall, abi?
You get it. God bless you. So capsule is there.
Oh,
So
so
capsule
capsule is like outer.
outer. And
Oh,
it's not all of
okay,
them that have it.
okay.
It's not all of them that
It's
have it.
not all because there's
Yeah.
something about vaccines when we get there, because it's been confusing my head.
Don't worry. I know what you are talking.
Okay,
I know
okay,
what you are
okay,
talking about.
okay.
We'll talk
Thanks.
about it now. We'll talk about it now. I know
Oh,
what you are saying. So
thank you.
shin,
Please,
streptococcus.
can you repeat what she said again? Sorry.
The capsule is the outer layer and is not present in all the bacteria. So a bacteria that has capsule is encapsulated. Do you get it?
Yeah, okay.
So streptococcus,
Dr.
Neisseria.
Babali,
Yeah,
just a minute. Sorry, don't be angry.
no wahala.
So there are organisms that have cell wall.
Okay.
There are organisms that have cell wall.
Mhm.
There are organisms that in their own cell wall, like another layer from their cell wall is the capsule. So some have the capsule, while some that have cell wall don't have the capsule, right?
Mm-hmm.
Yeah, yes, it depends.
Yes.
It
Yes,
varies. It
yes,
varies.
yes,
You know,
yes,
Dr.
yes,
Olatobi, there are different permutations
yes.
of these things. There are different. You need to take each individual organism and be sorting them out. Do you get?
Yeah,
Like
yeah, I get, I
look
get.
at E. coli. E. coli has capsule and is a gram negative. It has a cell wall, but the cell wall is thin. Do you get?
But it has a capsule.
But it has a
Even though
capsule.
it has a thin cell wall.
Even though it has a thin
Yes,
cell wall because
thank you.
it is gram negative. Do you
This
get?
example you gave helped it to stick more. Thank
Yeah. So that
you.
is it.
Yeah. So there's a difference between cell wall and capsule.
They are
Okay.
totally
Yeah,
different. They are
thanks.
totally different.
Thanks. Yeah.
Yeah. So you remember that all of them will have cell wall except chlamydia and except mycoplasma and ureaplasma. All of them have cell wall, but
Plasma. Yeah.
and mycoplasma. But the only difference that okay, is it gram positive or gram negative? Because it is the cell wall that will account for the gram positive or the gram negativity. So the one that has a thicker cell wall
Mm-hmm.
is a gram positive. The ones that has a
Mm-hmm.
thinner cell wall is a gram negative. Yeah.
Okay.
Well, where did you see all of them have cell wall? Which one are you talking about? All of which?
All of the organisms, we talked about it when
Which
we
organism?
see now. See, I mentioned it. I said peptidoglycan is present in both of them, and it constitutes the cell wall. But for gram positive, it is thicker. For gram negative, it is thinner. That is the basis for the gram staining. See, look at this. Look at this peptidoglycan here. Can you see this blue structure? See this blue structure.
Mm-hmm.
You can see
That's
like
Mm-hmm.
true.
three.
But they all have cell wall. They
They all have
all
cell
have
wall.
cell wall. That's true. That's
The
true.
ones
That's
that do not have
true.
cell wall, the ones that do not have cell wall is what? Mycoplasma
Mycoplasma and
and
uriplasma.
ureaplasma. It shows that gram
Yeah,
positive
that makes
has
sense.
thicker cell wall, while gram
Thick
negative
cell
has
wall. Yeah.
a thin cell wall. This is the reason why you can differentiate them with gram stain, because the one that has
Stain.
a thicker cell wall will pick up crystal violet and it will appear purple, while the one that has a thinner cell wall will not pick up the crystal violet very well. So you can decolorize it, and this remaining cell wall will pick the saffronin counter stain and it will appear red. Do you guys get it now? That was why I was trying to explain the
I
process.
get it, man. I now get it. I get it now. Dr. Lushala, can you look at the diagram
But
of the cell wall?
Dr. Wale, are you still recording?
Yes, I am. I am.
Okay.
Dr. Lushala, if you look at the diagram, you see that the peptidoglycan layer there are like 3 layers of peptidoglycan for the gram positive, and for the gram negative, it's just one peptidoglycan layer.
You can see
So
it here.
all organisms are divided into positive or negative. The ones that are not staining with gram are the ones that we brought out, the treponema, myco, and the legionella. But the ones that don't have any cell wall, cocon, is mycoplasma and uriplasma. Sure you get.
I get it.
Mhm.
Yeah. So these are the encapsulated organisms. That is the organism that has capsule. You said it that capsule helps to protect them against phagocytosis. I'm not sure Dr. Francois is here again, but we were talking about it. Capsule helps to
Yeah.
okay, Dr. Francois, you are there. That's good. I'm happy. So capsule helps to protect against phagocytosis. You can see it here. The capsule serves as an phagocytic virulence factor. Okay. What are these organisms? If you don't remember anything, shall remember strep pneumo, remember Haemophilus, remember Neisseria. They go together. And in addition to that, put group B streptococcus, because if I don't remember that group B streptococcus can, is encapsulated, you guys remember that we will not get that question of group B that we picked that day. Do you guys remember that we got one question that we're picking group B for the cause of the sepsis? When they were talking about beta hemolytic,
Yeah,
do you remember?
yeah, yeah,
The
yeah.
next question was after you have figured out that this is group B, the next question was what was the virulence factor? Do you remember? That was the next question. The
Is
next
it
one
is
was
it M protein, lipid A,
yeah,
capsule, and
Ah, yeah, I
the
remember the question.
options? Yes.
exactly. So the answer to that question was what? The capsular polysaccharide, because you have to remember that group B is encapsulated. Group B streptococcus is encapsulated. So you not pick M protein, you not pick protein A. We'll get there. We'll see all those ones that are using those ones. Okay. So I don't know what code to give us for this one. Mm. But I just believe these are the things that we might not need code for this one. Just remember Haemophilus, Streptococcus pneumoniae, Neisseria. Those ones they go together. Then group B strep. Then of course E. coli and Kleb, they are together. E. coli and Kleb. E. coli Kleb, encapsulated. Pseudomonas 2 and Salmonella. I don't know. The thing is, I believe that if you see it in question, you'll be able to remember that Pseudomonas is encapsulated. Like, you guys might. The thing is, with time, you will know. It will just be part of you. It will just be because you've seen it many times. Strep pneumo, Haemophilus, Neisseria, E. coli Kleb, Pseudomonas.
Shola will help us figure out a
Yeah,
mnemonic for it. Don't worry.
for
I
yeah,
trust him.
yeah,
Dr. Shola, we await your expertise. Thank you.
yeah. So now there's a point here. I think this is the point Dr. Olatobi was trying to talk about, and it says capsular polysaccharide plus or minus protein conjugate can serve as antigen in vaccines. Yeah, so we need to know about these guys very well. They test it a lot. Now, what they are saying is that you can derive antigen from this organism, this encapsulated organism. You just go to their, because one of the virulent property is a capsule. So you can just take part of the capsule, you take a polysaccharide, because we said that the capsule is made up of polysaccharide. So that means that if you are taking a portion of the capsule, you are taking a polysaccharide. So if you give, if of course, if you take a portion of that polysaccharide, that means you have a vaccine. So vaccine is like an antigen of a particular microbe that can generate immune response. We said it. Antigen is what you can produce antibody against. Now, that capsular polysaccharide, once you introduce it as a vaccine, it will not trigger a very robust immune response, such that it's because we've not done immunology, but I'll try to explain. It will not lead to production of memory cells. You will not form IgG, you will not form all the other ones. The only thing that you form is your IgM, and you will not form memory. And the process of even forming vaccine is useless, because the reason why you are vaccinating somebody is that whenever the person encounters that infection again, the person can have a response to it. When you are forming IgM number one is short-lived, is a primary immune response. When you form IgM antibody, they are primary immune response, and they are not long-lived, they are short-lived, and they don't have any memory component. Whenever the organism come again, nothing will respond to it. The only thing that forms memory is when you form IgG to that antigen. But when you form IgM to that antigen, you are not forming any memory cell. So if you give somebody the capsular polysaccharide only, just the polysaccharide, no protein is there, you package it as a vaccine, you introduce it. The B cells, guys, the B cells are going to recognize it, and they are going to produce IgM that will, respond to it. That is all. It is only IgM that will be responding to it, and IgM is not, it doesn't, build a very robust immune response like that. That is why capsular polysaccharide or polysaccharide antigen are not as strong as when you conjugate them with protein. Now, when you add protein to this polysaccharide antigen, you have added a protein that is complementary to that, polysaccharide stuff. Now, we'll call it a conjugate vaccine because you have conjugated the protein with the polysaccharide. That protein component can be recognized by T cells. Now, it is not B cells that is seeing it now, it is the T cells. The T cells will now stimulate B cells to produce IgM as well as IgG that is specific to that antigen, to that protein antigen. So the IgG will stay there. They will not participate in the primary immune response, that is the immediate one. They will stay. They will go and hide somewhere. By the time that infection is coming in again, the IgG will now mount the secondary response that will be very robust. They will clear it fast. That's the concept of vaccination. That's how a proper vaccination should occur. You should have memory cells existing following encounter of the first antigen. So immunogenicity can be enhanced when you conjugate the capsule antigen with the carrier protein, because you are going to have your T cells being involved. T cells is now going to stimulate the B cells to differentiate into your IgG and your IgM. Dr. Oluwatobi, does that make sense to you now, or you want me to explain again?
It makes sense to me. I think what was confusing me before was that so there is the non-typeable Haemophilus influenza, and then there's Haemophilus influenza virus. Do you get? And then there are like some people will get the vaccine. I think I'm jumping, but since you asked, so the people will get the vaccine and still have the same strain. So I was reading in UWorld and they were like, yeah, so they developed that one because of the capsular something. Do you get? So that's just the
Yeah.
part that was not clear. The whole capsule thing.
Yeah, so this is it. When you say the non-typeable hemophilus, we are going to get there. Non-typeable hemophilus is non-capsulated form of hemophilus. It is not form of hemophilus that is capsulated. That's another thing. There's a type that has capsule, there's a type that does not have capsule. So because we
Okay.
are producing antigen, you know that you are using the capsule as antigen. You know there is a capsule
Yeah.
that you're using. So that means that there's no antigen
Yeah.
for the non-capsulated one. So that means that you cannot produce vaccine against it. That is why they are
Against
saying
it.
that. That's why they are saying
Okay.
that majority of the infection from hemophilus that is dominating now are from the non
Non-type people one.
non-typeable or the unencapsulated ones.
Encapsulated one. So
You
you
get it?
can type it when it has a capsule.
Exactly.
Correct.
You
Thank
get it?
you.
You type it when it
Thank
has a
you.
capsule. Without capsule, you cannot type it and you cannot produce vaccine against it.
Vaccines against it. Okay. 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 Thank you
So
so much.
yeah. So the the way you can now take care of, because we've said it, encapsulated organism, the capsule is helping them, you know, escape phagocytosis. But how can the body now, just imagine the body is now infected with hemophilus. Does it now mean that, or strep pneumo, does it mean that we'll not be able to mount immune response? No, we will be able to mount immune response, provided that we have a functioning spleen. Provided that we have a functioning spleen. One thing I want you guys to learn here is that the macrophages are the ones that are involved in phagocytosis. You phagocytose, you clear them. But the macrophage cannot phagocytose something that does not express protein. They cannot phagocytose something that does not contain protein directly. No. Now, because of the fact that all these capsule stuff, they don't contain protein, it's polysaccharide. That means that phagocytosis cannot work against them. But how can we now clear them? What will now happen is that the spleen will now start releasing IgM and IgG, IgG predominantly. Now, by the time we are getting to, that is why you see that all these lectures, they are following each other. We should have done immunology before we are progressing to this lecture, so that we'll be able to appreciate some terms. By the time the spleen release IgG, IgG has two components. We call them let me just say FAB and FC. Now, FAB is the antigenic component. C is the part that the phagocytosis molecule, or the phagocytosing cell, let's say macrophage, is going to bind to. Now, get it again. IgG is an antibody. It contains two components, the FAB region and the FC region. FAB is where the antigen is going to bind to. That is, antibody is specific for a particular antigen. So the antigen that is corresponding to that antigen will bind to the FAB. Do you get it?
Okay, that's correct. Yes, yes.
So you have an antigen antibody complex like that. Now, in the FC region of that antibody, it can bind to another thing like a macrophage. Do you get it? So that means that once the macrophage is able to bind to that FC region of the immunoglobulin or the antibody, it can take care of all the complex. It will phagocytose all the complex like that. It will now take it to the spleen. Spleen will now destroy everything. So the way by which we take care of encapsulated organism is what? The spleen will release IgG that can bind to that, that capsular polysaccharide. So once the spleen release the IgG, they will bind to the capsular polysaccharide. The capsular polysaccharide will be binding to the FAB region of the IgG. Macrophages will now come and they will now bind to the FC region of the IgG. You can see that the macrophages are not binding to the capsular polysaccharide directly. They are binding
up at the lower part.
Exactly. So they are binding with the antibody serving as an intermediate. You can see. So they are binding.
So what is calling their attention is the antigen binding to the antibody portion, and then they will now come and go to the lower part and just chop and bind to it and chop off everything at once as a whole.
So that complex like that, the macrophage will try to swallow that complex like that and not take it to the spleen. Spleen will now mop everything up. Now, that is the way you can respond to encapsulated organism. So that means that anybody that has problem with the spleen and they cannot release the IgG and the spleen cannot mop up, even if they find a way to release IgG, the spleen cannot mop up the macrophage, the IgG, the antigen complex. There's no way they can take care of the bacteria. So people that are asplenic for any reason are predisposed to infection from encapsulated organisms. Do you get the reason why they now say they are predisposed to, infection from, people that have no spleen? That is the reason. Now, all those things that we have talked about, antibody being released from the spleen, binding to the antigen, the capsular polysaccharide, the macrophages coming into, that's the problem of opsonization. You opsonize it, antibody, the antibody will go and bind to the antigen so that the macrophages can engulf it. That is opsonization. And it is the spleen that helps us with that process. And what are the two implicating things? IgG and C3b. So I don't want to mention C3b is a complement protein. We'll be knowing opsonization proper later. So it means that anybody that doesn't have a spleen will be predisposed to infection from encapsulated bacteria because of that. And the three common organisms like that is the Shin, the strep pneumo, the H influenza, and the Neisseria meningitis. So they can give you a scenario of someone that has no spleen. They can tell you this person has autosplenectomy from sickle cell. They can say the person has ruptured the spleen either from trauma. They can say anything, or they've removed the spleen from any reason. They will now ask you, which of the following organism will the person be predisposed to? You just look for any organism in the option that is encapsulated. Most of the time, they will give you between these three guys. You just pick it and you go. So do you guys get it now? Does it make sense to us? So I know that it will be a little bit difficult.
Yes.
Yeah, I get it. It makes sense to me. Yeah.
By the time we are doing immunology, we'll get it well. Yeah. So, urease positive organisms. This is the next one. Urease positive organism. The code is still the P chunks. It's just like what they have here. P chunks. I know that we're already familiar with some of them when we're doing renals. We already know Kleb. We already know our staff. But what I just want us to know here is that Staph aureus is not here. It is Staph epidermidis and Staph saprophyticus. So The P chunks, the P is proteus, that's the baba, we already know that. Proteus is urease positive. Why? Because if something is urease positive, it can split urea to release ammonia and CO2. But don't be deceived. It's actually the ammonia that will predominate. Your CO2 will just disappear. CO2 is a volatile gas. It will just go to the airway and it will be excreted. It is the ammonia that will stay. So the ammonia is the one that will stay and it will raise the pH. So that is why organisms like Helicobacter pylori need this urease to function. Because if Helicobacter pylori can create an environment that has high urea, it can neutralize the acid around it and it can stay in the stomach. Stomach is highly acidic. It tries to produce ammonia through this urease so that it can dilute the acidic environment so that it can stay. In response to that ammonia, gastrin will be secreted. You guys remember that gastrin is an hormone that helps to elevate what?
Acid level.
Acid. You get it will help us to secrete more acid. So the ammonia will try to create a basic environment so that gastrin can fire and stimulate the gastric cells. The parental cells will start releasing HCl. That is the pathophysiology of how Helicobacter pylori can cause ulcer. Very important through this urease mechanism. So the acronym is P chunks. P is the proteus.
So, Dr. Babali,
Yes.
H. pylori creates a basic environment
Okay.
that now stimulates the parietal cells. Is it not the chief cells? The chief cells.
No, it's not chief. Chief is pepsinogen.
What secretes?
Parietal cells, acid and intrinsic factor.
So I was correct. Okay. So H. pylori
No, you are not
is
correct.
urease. No, I said it stimulates the parietal cells now.
I thought you said chief cells secrete pepsinogen.
I said parietal before, and I said chief because I wasn't sure of parietal.
Okay.
Sorry.
All right.
So you said H. pylori is urease, so it's producing ammonia. The ammonia is increasing the alkali nature of the stomach. So that stimulates the parietal cells to produce more from gastrin to produce more HCl, and that causes the ulcers. Is that it?
Yes, but gastrin has to come first. So in response to that alkalinity, gastrin is released from the G cells. So the gastrin now stimulates the parietal cells to release acid. Do you get it?
Okay. Thank you. The G cells in the stomach or duodenum, B?
Yeah, they are everywhere. They are plenty.
Okay.
They are plenty.
I get it now. Thanks.
Yeah, sorry. So that urea, that ammonia is also creating a shield for the helicobacteria to survive. You get it? So that it doesn't get destroyed by the acid too. You get? So so the P chunks produce because it can cleave off urease to form ammonia, and this ammonia can contribute to the formation of the struvite stone, which contains magnesium, ammonium, phosphate. So this ammonium component is coming from the urea, from the urease activity on urea. So they produce the ammonium, which will form the stone. That is why protease is notorious for causing struvite stone. Cryptococcus can also form ammonia. So what did we say about cryptococcus, Dr. Zuliat? What can you remember about cryptococcus? Okay, Dr. Zuliat is not here again. Dr. Francois, what can you remember about cryptococcus?
Hi. I think Cryptococcus is the one that we said is implicated in neurological infections.
Okay.
Right.
Meningitis.
Cryptococcus formans. It causes meningitis.
Encephalitis, okay.
No, meningitis. So meningitis. Indian ink. Indian ink.
Yes, Indian ink stain. What else?
Indian ink stain.
What else?
Musiramin. You said
Musticamine.
musiramin.
Right.
Yeah. Musikamin. Sorry. Musikamin.
Musticamine, Indian ink, meningitis. And it has, what is the virulent factor? That's what we are going to be seeing in the Indian ink.
Polysaccharide capsule.
Polysaccharide capsule, which will look circular. It has a very thick polysaccharide capsule, Cryptococcus neoformans. Yeah. So this is code chunks, P chunks, produce your Cryptococcus, Helicobacter pylori, the ureaplasma. So what do we say about ureaplasma?
It lacks a
He
cell
said it
wall.
No
doesn't have a
cell wall.
capsule.
It has no cell wall.
It
And
lacks a cell wall.
Exactly. So what about nocardia? What did we say about nocardia?
Nocardia. Did we talk about nocardia? We've not talked about nocardia at
We
all.
talked about nocardia.
But I know it
It's
is
also acid
acid
fast.
fast.
Acid
It's
It is
fast.
acid
acid
fast.
fast. It is acid fast. I
Hey,
mentioned it.
sorry, don't be angry. I'm very sorry. Very, very sorry.
Kleb, then Staph epidermidis and Staphylococcus. So Staph aureus is not here.
So you said Kleb is a lactose fermenter.
Yes, Kleb is the lactose fermenter.
Okay.
Yeah.
Staph epidermidis has biofilm. It forms a biofilm.
Fantastic. It
Okay.
forms a bio.
Slime layer.
These are the ways you guys can learn it. You see, you are joining it. That is the way. That's the way you can learn microbiology. That is it. Staph epidermidis, slime layer. So in which area? If you don't remember, it's fine. But do you remember the area?
Mechanical ventilators.
No.
Catheter.
Another one is mechanical.
Oh, that's pseudomonas.
That's
Catheter.
Catheters.
Prostatic valve, Abby.
Prostatic valve.
And catheter.
Yes,
Prostatic valve
catheters. I'm
is
so
catheter.
sorry. Yeah.
That's right. You guys.
Pseudomonas is the mechanical ventilator.
Yes. So
Yeah.
Pseudomonas is in cystic fibrosis. It colonizes the respiratory tree in cystic fibrosis patients, especially the elderly ones, the adults. It also forms the biofilms in the contact lens. So people that wear contact lens, they are predisposed to developing keratitis due to the fact that Pseudomonas can form biofilms within the lens. Then Pseudomonas can also cause mechanical ventilator associated pneumonia. That is it. Okay. So the catalase positive organism is very high yield. We meet it here, we meet it in immunology. We have to know it. Very easy. It looks very confusing. It's very confusing. That pathway is very confusing, but it's very easy. Once you can, pay attention to it.
Dr. Babawale.
Yes, Dr. Lishala.
Sorry. Did you read the Pseudomonas on urea is positive?
Urea is positive. No. We didn't.
Which one were you reading? The last thing you were saying? That cystic fibrosis something?
That's biofilm.
But you talked about Pseudomonas there, Abby.
Yes, I talked about Pseudomonas. So
Oh, okay.
Pseudomonas can form biofilm
Sorry.
in the respiratory tree of cystic fibrosis people. Sure you get.
Mm,
So it can
sorry, I
form.
was looking when you said Pseudomonas, I was not saying, is it part of urea is positive again?
No.
All right, thank you.
It is proteus, yeah. Proteus. So this is where you can now find Pseudomonas. Catalase positive list is very long, but the one that they commonly test is Staph, Aspergillus, and Nocardia. I've seen a question on Nocardia testing the catalase positivity effect of Nocardia. You must remember that. You must remember it. So catalase is an enzyme that degrades hydrogen peroxide into water and oxygen. Catalase, that's what it does. It degrades. Let me ask this question again. I don't know if you can remember, but it's okay if you don't remember it because this is the first time. So what group of organisms did we say they lack catalase?
These people now. Anaerobes.
Anaerobes. This is the mechanism. You can see. So they lack catalase because catalase helps to degrade what's it called? Hydrogen peroxide into water and oxygen. Then we also talked about the fact that they lack superoxide dismutase. Superoxide dismutase will help convert what's it called?
O2
Our superoxide to oxygen. So they lack those two enzymes. They lack those two enzymes. So catalase, because if just imagine they have catalase, that means that they will be producing oxygen now. Do you guys get? They will die. Do you guys get? They don't survive in area that has oxygen. So, and catalase, can degrade hydrogen peroxide into water and oxygen. And our superoxide dismutase can convert oxygen free radical to oxygen itself. That is superoxide to oxygen. superoxide dismutase. So they lack those two enzymes because they don't want oxygen to be around. Now, that's what catalase does. Degrade hydrogen peroxide to water and oxygen before it can be converted to microbial products by the enzyme myeloperoxidase. Now, I would love to take us to that. I don't know if I can just show us, but let you guys should just picture it. So, what happens is when you degrade um, catalase, when catalase degrades hydrogen peroxide into water and oxygen, this water will now combine with chloride to form hypochlorite. That reaction is catalyzed by myeloperoxidase. Now, hypochlorite is a very toxic agent to the cell. Any bacteria that has hypochlorite in it, it will die. Hypochlorite will kill it. So, people with chronic, people with
Dr. Babale,
yes, Dr. Lishola.
maybe you should speak with the image I just sent now.
Okay, let me see. Yeah, that's the image I was looking for. Okay, but I think it's best I show it because of I'm recording.
Okay, you can show.
Let me go. I don't
Okay.
want to go before, but I have to go. Let me look for that image. So I really want you guys. This is where we are going to stop. So
Dr. Baba, okay. Because I was about to ask them.
Where's my baby?
okay, so this is the image. Yeah. So this is a bacteria, being engulfed by the phagocyte. So you form a phagolysosome. That is, when a phagocyte fuses with a bacteria, they form a phagosome. So you join that phagosome with a lysosome so that it can degrade the bacteria. So how does the degradation process work? Now, the first thing is oxygen within the cell is converted. The oxygen within the cell itself, the environment is converted to superoxide. You can see oxygen being converted to superoxide. That reaction is driven by what we call NADPH oxidase. That's the Fox enzyme. NADPH oxidase oxidizes oxygen to um, superoxide. NADPH oxidizes oxygen to superoxide. The reaction is catalyzed by NADPH oxidase. The next thing is this superoxide. It is converted to hydrogen peroxide. That reaction is catalyzed by the superoxide dismutase. So to take it to hydrogen peroxide, the hydrogen peroxide will be acted upon by catalyst, degrading into O2 and water. Or you can incorporate chloride into this hydrogen peroxide and convert it to hypochlorite. So this reaction 3 is catalyzed by myeloperoxidase. So the first one, oxygen is converted to this guy, which is superoxide by NADPH oxidase. The superoxide is converted to hydrogen peroxide by superoxide dismutase. The hydrogen peroxide is combined with chloride and the reaction is catalyzed by myeloperoxidase to form hypochlorite. Hypochlorite, degrades the bacteria and the bacteria dies. Okay, so we said that organisms now deficiency of this guy, this NADPH oxidase is called chronic granulomatous disease. Why? What are we saying? When a cell is um, deficient of NADPH oxidase, that means that there's no way the macrophage or the lysosome can generate hydrogen superoxide. They cannot generate this first organism. They cannot generate this first molecule, which is the superoxide. So that means that there's no way they can eventually generate hypochlorite to degrade the bacteria. Now, what now happens is that there are some bacteria. This bacteria itself can release hydrogen peroxide. Now, the hydrogen peroxide, that means that if this bacteria releases hydrogen peroxide, you have bypassed this first step already. So that means that you don't need that first step. So the hydrogen peroxide can combine easily with hypochlorite, and the hypochlorite from the macrophage, and they will form. Sorry, the hydrogen peroxide can easily combine with the chloride from the macrophage, and they will form the hypochlorite, and they will use the hypochlorite to degrade that bacteria. Do you guys get what I said? Now, look at a condition. Someone has NADPH oxidase deficiency. If that person has NADPH oxidase deficiency, we are saying the person can develop chronic granulomatous disease. Why? Because the person will not be able to kill off bacteria. So the bacteria will be accumulating, and the macrophages will just be forming granulomas, but they will not degrade it at the end of the day. So the inflammatory response will be continuous, leading to formation of chronic granulomas. Why? Because they cannot degrade the bacteria. Why can't they degrade it? They lack NADPH oxidase. What is the function of NADPH oxidase? To generate superoxide. If they don't have superoxide, that means there's no way they can form hypochlorite that will degrade the bacteria. Do you guys get the reason why NADPH oxidase leads to chronic granulomatous disease? Let's settle down first. Do you guys get that?
Yes.
Very good. Now,
Yes.
if someone lacks NADPH oxidase and they cannot generate hypochlorite, there's a way they can generate the hypochlorite. How? Ask me how. They can generate the hypochlorite because bacteria themselves releases hydrogen peroxide from their own metabolism. They release hydrogen peroxide, and since the problem is just conversion of O2 to superoxide, they have chloride within the macrophage. So the chloride will just incorporate with the hydrogen peroxide from the bacteria, and they form hypochlorite to use to kill that bacteria. Do you guys get that? Now,
Which one has
Yeah.
peroxide? Which one did you say has
Sorry.
peroxide?
bacteria release the hydrogen peroxide, the bacteria themselves. You know that this hydrogen peroxide normally should be formed by the macrophages that are phagocytosing.
Exactly.
You get it? But since
Okay.
they don't have NADPH oxidase, guys, this thing can be very confusing, but just follow me. Since they don't have NADPH oxidase, that means that they cannot form um superoxide. So that means that there's no way superoxide will form hypochlorite, which is HOCl. Do you guys get it? So that means there's no way they can degrade bacteria. So if they cannot degrade bacteria, that means that they will have a chronic inflammatory response. You get, because they will keep responding to that bacteria, but they will not be able to degrade it. So in the process, they will just keep forming granuloma, just like someone that has a TB that cannot degrade the TB. Do you get it? That's why they call it chronic granulomatous disease, because of NADPH oxidase deficiency. But now, why they are not peculiar? Why they are not peculiar with the fact that it is only organisms that are catalyst negative that develops, chronic granulomatous disease in someone that in a macrophage with NADPH. I'm saying too many things. Now, they are saying it is only infection from catalyst negative organism that can lead to develop of chronic granulomatous disease, even though that person has NADPH oxidase deficiency. If that person doesn't get infected with an organism that is catalyst negative, he will not develop that condition. I will explain why. He will not develop the condition because we've said it that bacteria themselves can release hydrogen peroxide. So if the bacteria is releasing hydrogen peroxide, that means that you have, chloride already in the macrophages that can combine with the hydrogen peroxide to form HOCl that will degrade that bacteria. So if you can degrade the bacteria, there won't be any chronic granulomatous disease. Do you get that? Do you guys get that? I will come to,
Yeah,
Dr.
I
Babawale.
get that. But the catalyst part
Babawale,
is the part
don't
that
worry.
what me I'm not
because
getting.
I've not gotten
Let
see, doctor, see, I've not gotten to the catalyst. I just want you guys to be following that thought. I will explain
okay, we
the
are still with
catalyst.
you.
okay,
We're still with
but
you.
but let me tell you what I'm not getting. What I'm not getting is that
Okay.
you said they are afraid of organisms that are catalyst positive, Abi.
No, I did not say they are afraid though. I did not use the word afraid.
Okay,
I said
okay,
it is when there is infection by a catalyst negative organism that they develop this chronic granulomatous disease. I want to explain why. You see,
okay.
the fact that someone has NADPH oxidase deficiency in their macrophage doesn't mean that they will develop chronic granulomatous disease. No. They will only develop chronic granulomatous disease when they are infected by, when the macrophages are infected by catalyst negative organisms. Right.
I don't get that statement.
Okay.
I don't get that statement.
Now let me go back. I'm coming. I just want to show us this.
Because what me I'm thinking is that uh for us to I feel that the peroxide
Uh-huh.
the peroxide,
Okay.
I mean, number one, why would any organism wants to provide peroxide to kill itself?
No,
That is number one.
you see, they are not generating that peroxide to keep. Peroxide is one of the metabolic byproducts from bacteria. Do you get it? That peroxide is actually converted to a killer because the macrophages have chloride within them that can easily combine with the peroxide to destroy the bacteria.
Okay, okay.
So if now what so, so what I'm supposed to say, okay, I think I get where you guys are missing it. So I said by catalyst negative, so it should be catalyst positive. So if something has catalyst, that's
Yeah,
when
I think that's
yeah.
what I was like.
Okay,
Yeah.
okay,
Well,
sorry
that
about that.
scattered my brain. I just
Sorry,
wanted you to have like a
Honestly,
thing.
sorry.
I
So
think that's it.
if they are infected by oh guys, I'm so sorry. So if they are affected by a
it's
catalyst
fine.
positive organism, that's when the problem starts. Why? Because if they are affected by a catalyst positive organism, the even if the bacteria is releasing the the hydrogen peroxide, the bacteria will do its own metabolism, it will release hydrogen peroxide. But the fact that they are catalyst positive, they will cleave off that peroxide. They will cleave it off with their catalyst. So there won't be any peroxide for the macrophage to use to destroy the bacteria,
To form hypochlorite, yeah.
to form hypochlorite, to form the bacteria. So that was what I'm supposed to do. I'm so sorry about that. Sorry. I didn't know that I was saying the other way around is money. So that's why they say that people with infection, if someone with NADPase oxidative deficiency get infected with a catalyst positive organism, they can develop chronic granulomatous disease because of the fact that okay, they already have a challenge. There's NADPase oxidase deficiency. There's no way they can generate hypochlorite. There's no way they can kill the bacteria. But the only way they can kill the bacteria is when the bacteria does its own metabolism and is releasing hydrogen peroxide. So the hydrogen peroxide will get combined with Cl to form hypochlorite, and they will use that hypochlorite to destroy it. So there won't be
Mhm.
any CGD. There won't be any CGD. But when that bacteria has a mechanism of cleaving its own hydrogen peroxide, CGD will develop. CGD will develop. So I think we are on the same page now, guys.
So yes, so they are
Yes.
safe with catalyst negative, Abi.
Yeah, they are
They are
Yes,
still with
still
catalyst
we are
negative
still with catalyst
organisms.
negative. Yes, yes. Thank you, Dr. Luchola. So let me see if we can just. I think that's the last point there. I just want to see so we can just close this meeting. So that you guys can rest for. Yeah. So catalyst degrade hydrogen peroxide to water and oxygen before it can be converted to microbial product by so before it can be converted to microbicidal product like hypochlorite by the myeloperoxidase. Yeah. So people with CGD, which is NADPH oxidase deficiency, have recurrent infection with certain catalyst positive organism. You know, catalyst positive organism like these guys. So the code is just places. Once you know places, you've known a lot of them. Places, the P is Pseudomonas, the L Listeria, the A Aspergillus, the C Candida, the E coli, the S Serratia, the last S staph. So all the staph are here. Unlike Uris, that staph aureus is not there. Then uh you are left with the two B's, one H and one N. You already know nocardia, one of the N. Nocardia should always be in our mind. And the two B's, Bordetella pertussis and Bacchidoria sepacia. So these guys also notorious in people with cystic fibrosis. It's very common in them. Cystic fibrosis, Bacchidoria sepacia, Bordetella pertussis, Helicobacter, and nocardia. So that's it. Then places. Then remember the places. Pseudomonas, Listeria, Aspergillus, Candidiasis, E. coli, Staph, and Serratia. I said it without looking at it because this is not the first time I'm reading it, so I've read it a lot of times. So with time too, it will stay. You guys, once you look at it often, you will remember it, even without remembering the code. Once you just mention, you remember that okay, it's part of the Listeria, particularly Nocardia, Aspergillus, and Staph. They commonly ask those ones. Nocardia, Aspergillus, and staph. Yeah. So we are ending this meeting now.